Hello everyone, welcome to my presentation entitled Strain-induced Differentiation of Mesenchymal Stem Cells. My name is Khalil Moussi. I am PhD student in the Electrical Engineering Department at King Abdullah University of Science and Technology. In this presentation, I will start with the motivation. Then I will present to you the concept and design of the electromagnetic system used for cells differentiation. After that, I will show the simulation results of the strain distribution and how we correlate these results to the signal of the cell's differentiation. Mesenchymal stem cells are multipotent stromal cells. They are derived from several tissues in the human body, as you can see in this cartoon. More importantly, they have the capacity to propagate in culture while retaining their differentiation potential towards several lineages. Regardless of the tissue of origin, all human mesenchymal stem cells can differentiate in vitro into nerve cells, cardiac cells, osteoblasts, chondrocytes, and myocytes, among other cell types. However, directing the fate of human mesenchymal stem cells toward bone formation, for example, is a promising approach in regenerative medicine related to bone diseases. Different approaches have been applied to improve selective osteoinduction and consequently to reduce the time required to achieve differentiation. However, the main drawback of most of these studies is that they use molecular products like growth factors to exhibit specific outcomes. In this work, we investigate the effect of external mechanical forces without the addition of any external cues in order to understand the actual force needed to accelerate differentiation of human mesenchymal stem cells towards specific lineage, osteoblasts, for instance. Now let's move to the concept and design. As you can see in this schematic, the experimental setup is composed of a solenoid with a membrane holder on top of it, as you can see as well in this photograph. Human mesenchymal stem cells were cultured on top of an elastic membrane made from PDMS material. A permanent magnet disc was attached to the bottom side of the membrane, which was magnetically actuated to induce a periodically varying strain. As you can see in this photograph, the setup was placed inside an incubator, and the solenoid was connected to an AC amplifier in combination with a function generator, and the membrane vibrated at a frequency of 24 Hz. Now let's move to the simulation part. So basically, it's a finite element modeling simulation using COMSOL software. The membrane and magnet, they have both an axis symmetric geometry, so the model for the simulation can be simplified as follows, as you can see in this 2D schematic. For the maximum deflection of the membrane, it was measured and extracted from the actual experimental setup, which was 3.5 millimeters in the positive and negative Z directions. This is an animation of the strain distribution along the membrane for a deflection of plus minus 3.5 millimeters. So we can see the strain distribution along the membrane. This is an animation of the strain distribution. Here we have an animation of the strain distribution along the cross-section of the membrane for a deflection of plus minus 3.5 millimeters as well. So here we focus on the positive deflection of the membrane when it is deflected with 3.5 millimeters in the positive direction. We have a 3D view of the strain distribution as well as a cross-section view of the strain distribution. Now, as the cells were seated in the top surface of the membrane, so we are interested of the strain profile for this upper surface of the membrane. This is the strain distribution in the positive deflection of the membrane. And this is a schematic view of the membrane. So we noticed from the strain distribution 4 different zones. Zone 1, this is the upper surface of the membrane where it is attached to the magnet. So basically here, the membrane will not stretch, and that's why we have 0 strain. Zone 2, this is the upper surface directly on top of the edge of the membrane. And when the magnet is deflected in the positive direction, we can have here the maximum strain with about 12%. Zone 3 is the area of the membrane between the magnet edge and the surrounding of the membrane holder edge. And we found a strain of about 2 to 3%. Finally, in the surrounding of the membrane holder, we have a small spike of 2% strain. Now, for the negative deflection of the membrane in the negative Z direction, minus 3.5 millimeters, we have a 3D view of the strain distribution as well as the cross-section view. We can see in this magnified view, as well as in this 3D view, there is a higher strain in this corner, but this is in the bottom surface. However, the cells were seated in the top surface of the membrane, and we will investigate the strain distribution. So we have, again, similarly, 4 different zones. Zone 1, where, as we said, the membrane, where the magnet is attached to the membrane, and we had, the membrane can't stretch, and basically we have 0 strain. We have a spike of strain exactly on top of the magnet edge with 4% of strain. The region between the magnet edge and the membrane's holder edge, we have a strain of about 3 to 2%. And here we noticed a second spike of strain of about 4%. When it is deflected, the membrane is deflected in the negative direction, we have in zone 4 another spike of about 4%. This is the combination of the strain distribution for both deflections of the membrane in the positive and negative directions. Now let's move to the final part related to the cell's differentiation signal. As you can see in this graph, this is the Alizarin S red signal measuring the distribution of human mesenchymal stem cells differentiation into osteogenic cells after 14 days of incubation under constant vibration. This result is 7 days earlier than the normal differentiation time using chemical methods. It is possible to detect osteogenic differentiation of human mesenchymal stem cells in zone 2 and zone 4, and that's where we have the region or the highest strain distribution. Similar results were obtained after 17 days of incubation. We can detect the osteogenic differentiation of human mesenchymal stem cells in zone 2 and 4, and that's where we have the higher strain. Finally, the results at 21 days showed a saturation of osteoblast differentiation in zone 2, 3, and 4. So indicating here that there may be a positive feedback effect of those cells differentiated in zone 2 and zone 4 to those cells in zone 3. Here we can see optical microscope images of the human mesenchymal stem cells from the center to the border, to the outer border of the membrane, where human mesenchymal stem cells were grown under constant vibration and consequently stained with Alizarin S red. To sum up, human mesenchymal stem cells were cultured on a magnetically actuated membrane to induce periodically varying strain. So the strain distribution across the membrane was calculated by finite-element modeling simulation, and we have showed strain-dependent differentiation where the highest region of strain resulted in the most accelerated differentiation. And finally, osteogenic differentiation was achieved as early as 14 days of incubation time. Thank you for watching my presentation.